why don't we get started? So I just started the recording button, so this is being recorded. So welcome to our eighth webinar entitled Targeted Therapy, and Jane will be giving this talk. And Jane is a long-time personal friend and consultant for CISN, and has actually helped us all along the way, and has been the key developer along with myself of the website. So she helped a lot on the research section, helped with the index, put together the index in fact, and then added to each section a lot. And so Jane, if you want to go to the next slide.
Oh, sorry. Oops.
Yeah. So we'll just go back to the mission statement for a bit. Just the same as always. We're focused on bringing stakeholders together, and part of that is through training and education, and part of that is this webinar. So Jane and I developed the content for this section of the website and also the PowerPoint. Jane is going to give you a little bit of her background. So Jane, why don't we hand it off to you? Thank
okay. Well, thank you. And as Peggy has already mentioned, I would really welcome this to be as interactive as possible. I know many of you, and you know me, and I'm a little bit shocked because I think some of the people on this webinar could do a better job than I can. But we'll learn from each other, which is why I like to do these things. And the slides were actually done quite a while ago, and they were, I think, meant for a very broad audience. So I will probably really speed through some and really go into a lot more information about some of the others. I am a 30-plus year breast cancer survivor and have a really interest in the research and in clinical trial design. I've been involved in the Alliance, where I know some of you, and many other cancer research projects. I'm semi-retired and spend more and more time doing more and more really interesting and very rewarding activities. So that's who I am, and again, feel free to ask questions, and there's a little bit of a delay from the time I press to go to the next slide till we get it. So I've been pressing the next slide for a while, so we'll have to work with that technology, but that's what we wanted to find out. What are our objectives? So our objectives is to learn what's meant by targeted therapy, become familiar with many of the approaches, and understand the complexity and magic bullet. And the topics that we're going to cover when the slide decides to come on. Okay, whoops. Overview of the research process. I'm going to really zip through, and those people who want us to spend any more time on it than I plan to, please let me know. We'll talk a little bit about what targeted therapy is, and I will try to relate it to some other terms that I think are sometimes used interchangeably or in different ways. And then we'll go through some examples of a bit on angiogenesis, which is probably has not taken off that much. I'm going to go through the immune therapy a little bit more than we originally planned, because I think that's the most exciting area in cancer treatment now. And we could easily have multiple lectures just on that. And I will do very little on gene therapy, which in my mind has not really taken off. And then we'll do a summary, and we'll have time for questions, but I hope that by then we have all the questions answered. So I am still waiting for the slides to move. I think, okay, so overview of the research process, and is anyone else getting a lot of static? There, cleared up. So, Peggy, if you control the slides, might they go faster? Because I'm having.
You want to be on introduction?
I guess I just, I'm just not sure how to get them moving faster. But we'll get started. So, background is, I think anyone who's on this webinar knows that cancer is a problem. It's been a problem to us. We've all been personally affected by the disease some way or another. And there have been significant progress in cancer. I often get kind of discouraged because I think that the progress hasn't been nearly fast enough, and because we seem to learn more about things that we didn't realize we need to know. As we're learning things that we know we need to know, we're learning more things that we didn't know we need to know that we do. I think Rumsfeld said something like that. So I'm waiting for a slide just in case you're wondering.
Okay, there you go.
So we know that new treatments come from research. We're all involved in it one way or another. We know that this war on cancer has been going on for about 45 years, probably older than some of you, but not older than me. Lots of money has been invested, both taxpayer dollars, not-for-profits, and drug companies. And we've learned a lot, and we know a lot more about the science than we did certainly 45 years ago, but we still haven't solved the problem. We also know that there are multiple pieces to the puzzle and multiple scientists, and this slide kind of shows some of the people that are involved, and I think increasingly the science is becoming multidisciplinary and team science, and actually that is one significant change that I've seen in my time involved with this. We also have talked a lot about over the years going from the bench, which are typically tissues, to animal studies, which are, quite a few people have been able to cure cancer in mice, but then when they go into clinical trials, doesn't always work. Successful trials lead to actually change in practice, and it's cyclical, and increasingly people talk to each other in these different areas, which I think is good. But the bottom line is that science is very incremental, lots of small experiments in the basic science, and lots of phase 1 and 2 trials that never get on to phase 3 where there can be real changes. But breakthroughs often happen in strange places. It's a very interesting book I can recommend to some of you on the serendipity of science, and also new technology plays a key role, and that's certainly been true in cancer research and in targeted therapies. So most don't get to clinical trials, and as I said, many that get to phase 1 are too toxic or not effective and don't move on. We all know how long clinical trials take, and there are many of them, and we're involved in the cooperative groups or NCI, so I assume, and/or you've had some earlier sessions, so I assume all this is pretty much old hat to you, but as I sort of present the bottom line, I'm going to pause and just see if anyone has any questions at this point that they would like to raise, or comments, because your comments will inform all of us. And also, the audio is you're not hearing
I think there's a lot of static right now.
I've been getting a little static too, on and off.
So, Jane, I'm going to mute people because that takes care of the static,
Okay.
and then we'll unmute again later.
Okay, but please do, even if you are mute, use the question field and ask questions, or I think you can raise your hand or something. I get lonely when I talk too much. So we're trying to move ahead to what we mean by targeted therapies. And I guess I'd like to spend a little bit of time here talking about terminology, because we have the word targeted therapies, we have the word personalized medicine, we have the word precision medicine, and in many ways I think they're getting at the same thing. As you'll see, I think targeted therapy was probably the initial term, and the idea is that we try to do things that will get at mechanisms in the cancer cell that will differentially kill the cancer cell rather than other cells. And what basic chemotherapy, as opposed to targeted therapy, is therapy that sort of just interferes with the cell cycle, whereas targeted therapy usually tries to target a mechanism that's more specific to the cancer. And as we have learned that there's such a variety of cancers that perhaps not even any two cancers are alike, let alone any organ site cancer. I think we even know there's a lot of heterogeneity within a single patient's cancer. So we started saying we want to personalize the medicine, and for more than 15 years that term has gone on, and the idea was you would find out about the particular person's tumor and treat them. Not only would you be targeting mechanisms in cancer, but you would be doing it specifically to the person. And in my mind, the word personalized medicine took on a lot of things besides the cancer that had to do with the person and their values and things of that sort. And I think that this is a little bit cynical, as some of you know I can be. I think some of the scientists got a little bit concerned that they might be promising to take care of things besides the cancer or take into account. So now we use the word precision medicine. Sounds good to the scientists anyway, and I think it allows them to focus on the cancer as opposed to the whole individual. Just my cynicism, perhaps. And the word precision medicine, I think, is often meant specifically to talk about the genomics of the cancer cell, whereas targeted therapy might also focus on some larger things in the microenvironment. And we'll hear about that in some of the coming sections. So I don't know if anyone has some reactions to my very personal definitions there, but
this is Patty.
Yes,
can you hear me?
Yes, I can.
Okay, good. Because at the conference, we were just out at ASCO breast meeting, Dan Hayes talked about precision medicine as making personalized medicine more precise. That was his definition. I thought it was
As some of you know, Dan and I both live in Ann Arbor and see each other regularly, and we like to debate each other regularly. So I will take him on that. I think what that, and he probably does mean, we're not going to take into account silly things like patient values, but thank you for sharing that.
Well, that's how you get around to the genomics, how the genomics is playing into that, whereas personalized medicine involves everything, including public health type things. Yeah. He had a good presentation.
Yeah, he's a very good speaker, and he's a very good guy, and he's very advocate friendly. But he does, as some of you might know, I love a good debate, and he's always ready and willing.
He is.
So I'm still having trouble moving to the next slide.
And so, Jane, on that question of personalized versus targeted versus precision,
yes.
When you Google personalized medicine, there's the primary care people have really jumped on that, and they actually now really want to deliver personalized medicine. And for them, it's clinical symptom management, it's environmental exposures, and it's laboratory or genomics or the kind of things that oncologists think about. So if you come out of the cancer realm, personalized medicine really does play an exciting new role. And you're right, in precision medicine, they're sort of stuck on the omics.
Right, and I think it probably primary care versus researchers, but also, I mean, I think the trend over time is going to be to call it precision medicine. As we now have the precision medicine initiative that Obama is funding.
Right. So I did have a question from Dorothy, and she said, whenever we get to some place where you can include what advocates would need to know or be able to convey to their constituents, that would be good.
Okay, and Dorothy, I think that's a good question for you to ask, and I think, to some extent, we as advocates, I think, try to inform ourselves as much as possible, and who you need to tell, what constituents you need to tell what, probably varies on the particular person, and you will probably figure out how to tell it to your community and individual people you talk to best by knowing them. But if some things pop up in my mind relevant to that, I will try to mention it. So as I already sort of alluded to, traditional chemo is typically goes after the function of the cell and the cell cycle and reproduction of the cell. Targeted is something a little more specific to either the cell or the environment. So now we have some examples of things that we might target, and a number of these, are really genomic, particularly the mutations and signals. But then when we get to things like blood vessels, we're talking, and the immune system, we're talking about things that are in the environment. So what makes cancer different than other parts of non-cancer cells? And these are sort of the critical pieces, really the breakthrough in the last probably 20 or 30 years, and then much more advanced after the mapping of the human genome, is to recognize that cancer is really genetic abnormalities, and these cells divide too rapidly. They're developmentally immature. They send out signals that generate blood supply and help them continue to grow and then metastasize through that blood supply. So the treatments are going to try to kill these things, and we're going to try to do it with minimal harm to the body. We do that with surgery, which is local, cut it out, radiate it, burn it out, and whole body therapies, which are traditional chemo. Hormone therapy, I think, really is a targeted therapy, but we put it in a different category because it's not quite as new and sexy as some of these newer targeted ones. So targeted therapy could be more effective, and this, is sometimes true and sometimes not. Because it goes right to what's really special about that cancer. So you're hopefully not going to get all kinds of side effects, and the drug is not going to be getting to other parts of the body. In some cases, this has actually turned out to be quite true, and on average, targeted therapies have turned out to be less toxic than chemotherapy, but it's definitely not not always the case, and in fact, many attempts to new targeted therapies don't get far because of toxicities. As an aside, and this is something that I think it confused me for many, many years. Drugs always have two names: a brand name and a generic name, and the example is Herceptin and trastuzumab. Unfortunately, when you're doing clinical trials, you have to use the generic name, and that really can confuse a patient, particularly if you're doing some standard of care piece. And sometimes the company brand name really gets picked up, and sometime for quite a while the generic name is what's used. But if you know that, it will help when you start talking about drugs. They also sometimes produce different drugs that are aimed at the same target. So Herceptin is aimed at the HER2 expression, and there are other drugs that are aimed at other parts of that pathway, or even the same pathway, such as lapatinib and pertuzumab. Another thing that's important for us to to help us sort of orient ourselves is that there are small molecule drugs and what are called monoclonal antibodies. And the small molecules, number one, are small, and they're biologically active, but they're not proteins, and they often end in an IB. Whereas monoclonal antibodies, and we'll talk more about that when we get to the immune system, are big molecules. They're proteins. They have lots of parts to them, and they often end in MAB, like trastuzumab. There are exceptions to all these rules, but I think you'll see that. Targeted therapies can be either. So Gleevec is sort of one of the poster children for targeted therapy. It's a small molecule, ends in NIB, and it's tailored to inhibit some tyrosine kinase enzymes, and it actually has been sort of a miracle drug for GIST, a particular type of gastric cancer that is in relatively small numbers of patients, but it has had remarkable and often, but not always, long-term results. Tumors often actually develop resistance, and there are some newer drugs that try to overcome the resistance, but it is a therapy that has had major impact on GIST, in part because GIST has a very kind of consistent mutation that it always goes right after, and there's an interesting book about the development of the drug and trials and tribulations that is good popular reading. Let's see if we can move this forward. Okay, so there are a lot of other examples, and we have some other small molecule drugs, Iressa, Tarceva. These are targeting the EGFR oncogene, which is actually present in many cancers, particularly, especially of the cell lining, the organ lining, which are epithelial cancers, but not all. Actually, a number of these drugs have been approved in one cancer, and then when they're found that other cancers, or at least subsets of other cancers, have the same mutations or overexpression, and they can be used. So Herceptin, which has had a huge impact in breast cancer, has also been found in a limited number of lung cancers and other cancers, and when those are found, they're often, but not always, the drug is often, but not always effective. And that's what we sort of mean by targeted therapy, or precision medicine, where we're looking at the individual cells. So when we talk about Herceptin, this slide actually shows how it actually impacts, and any antibody, what it does is it gets a hold of an antigen or some kind of thing in the cell, and that's the way it has its impact, and it blocks whatever that was going to be doing. And some people will call any antibody, that is, any drug that ends in an AB and that actually hooks into a receptor, that some people think of that as immune therapy because it's using a basic mechanism of antigen and antibody to do its work. But I think it would be best to reserve the word immune therapy for therapies that actually go about their work by targeting not something on the cancer cell, but something that's part of the immune system. But we'll talk about that more later when we get to immune targets. So here we have a whole bunch of targeted therapies that have actually been approved, that we listed here what their targets are, and most of them have been approved first in a single disease, and then trials go on to see whether or not they might be effective in other organ sites, and particularly for small subsets of patients that might express these targets. We'll talk about some of them more in a minute. So just to summarize, traditional chemotherapy goes after all the cells, and they're rapidly dividing. Newer targeted therapies only go after the cells that have these specific targets, typically the cancer cells. Typically, with traditional therapy, we use what's called a maximum tolerated dose. And we do phase 1 trials to figure out what that dose is. We keep increasing the dose with different patients until patients have really severe responses, and then we go back a little bit and give less, because it seems like, I mean, it's sort of this delicate balance between killing all the cancer but not killing too much of the other important stuff in the patient. It turns out that in targeted therapies, that same paradigm doesn't hold. There seems to be an optimal amount to kill the cancer cells, but we don't really, that may or may not be all that related to toxicities. Unfortunately, we don't really have a very good way of figuring out what that optimum is, so we often just go for, something that's either has, a lot of side effects, or when we keep increasing and we don't see anything, we'll stop at some point. And it's an area that I think we should be doing better at, and as advocates we might be asking those questions. Another actually issue that I think we should be asking about is, even with chemotherapies, people metabolize drugs quite differently, and they do what calls pharmacokinetics to understand how the drug gets and stays in the system. And they dose, for the average patient, and there's a lot of variability. And I think if we dosed individually, we probably could do better with the therapy and do in efficacy and lower the toxicity. So a guy at Michigan, for example, is looking at the toxicity of neuropathy in Taxol types of drugs, and he found that although there's a standard amount of drug to give to a patient based on their weight, how the patient metabolizes that and how much is in their system for how long varies quite a bit. And the longer it's there for the very same dose, but the patient that has it around for a long time is much more likely to get neuropathy. And that's not very pleasant toxicity, and sometimes it is not reversible. So you would like to not have that happen. On the other hand, there are other patients that don't get neuropathy and may well tolerate more and need more drug to kill all the cancer because they just don't have it around their system long. So in some cases, not all, the toxicity and the efficacy could be related. It means that the drug is having an effect. But sometimes, patients that don't have any side effects from the Taxol may actually tolerate very well a higher dose and may need that to kill the cancers because they metabolize it rapidly. So that was really an aside just of interest to me. We also have traditional therapies virtually always are given by IV infusion. Many of the targeted therapies are given orally, which has quite a number of advantages and some disadvantages for patients, like sometimes the reimbursement is not as simple, and also having patient compliance is sometimes an issue. So when you're reviewing trial concepts, it's something to ask about and think about. And if it's going to be an oral drug, there are things that can be done to help the patients, especially if it's they're complicated multiple drugs. You can use pill cases and things like that to help patients know what to take when. And finally, chemotherapy is very often very toxic, although we now have many supportive drugs to decrease that. And the targeted therapies were believed to be less toxic, but probably that was a promise that was not totally realized. I believe that in chemotherapy, nausea is one of a very important toxicity. In targeted therapies, that is less true because the targeted therapy doesn't get to the gut necessarily, but on the other hand, there's often quite severe skin rashes. In fact, when we talk about some of the particular ones, we'll mention that. So the bottom line is that targeted therapies may have fewer effects, which would lead to better quality of life, and they sometimes can actually fix abnormalities in cells and have long-term durability. Although we've been finding that most cancers end up having various mechanisms to become resistant to the therapy, and you need often to have alternative approaches. Sometimes they can be administered orally. As you know, many of these new targeted therapies cost $1 zillion. And rather than decreasing over time, they seem to be increasing not only from therapy to therapy, but the therapy that we use for GIST, for example, has actually increased in price significantly since it was first introduced. And as I mentioned, mutating cancers are often an issue, and therefore we often try to use combinations, which add toxicity. So when the slide decides to change, we'll start talking about a couple of the very specific targets that some of these therapies go after.
So Jane, we've got a few questions from Jim.
Okay, Jim. They don't show up on my screen, but I'd love to hear from you.
So his was, and you mentioned it a little bit on the last slide, that a major concern with target therapy is that the target is constantly moving. New clones emerge, and the target or pathway may no longer be a significant driver. Right.
So, Jim, you're right on, and I think that's one of the sort of disappointments. Although drug developers are learning more about two things. One, what those resistance mechanisms are likely to be, and trying to design drugs that hit multiple things at the same time, or multiple pockets in the same tumor or protein. And or having second and third generation drugs that then look at the address the mutations. But it's difficult. A lot of times the use of combination therapies is thought to reduce that issue, because it's not totally clear whether the cancer is mutating so much, or the tumor was very heterogeneous to start with, and we're just systematically killing a subset of the tumor. So it could just be that there were always a variety of mutations, but as we kill one type, the other becomes predominant. And if we use combinations where we have a better chance of killing more types right from the beginning and not having that kind of resistance. But that raises a whole other question about whether we're better off doing combinations of treatments or whether we're better off using them in sequence. And I don't think one size fits all, but another pet peeve of mine is that we really never do those kinds of trials. Most of the trials that we do are just looking to get a new drug approved. So Jim, do you have some more questions, and or was that a decent answer?
Yeah, well, so the second question is, instead of MTD, he's suggesting that we use MED, which is minimal effective dose.
Right, but how we find that is very, very hard, because we use phase 1 trials where you see toxicities right away, and you know when to stop. And you know, if your effective dose is going to cure the cancer, for example, you would you'd have to go for a long time to know that that was an effective dose, and then probably people would say, well, it's never going to be effective in anyone. If we give a little more, maybe we could do better. So, theoretically, that's exactly right, but how we get there is something that maybe, Jim, you can help us figure out.
Okay, that's it for now.
Okay, so we're going to turn to angiogenesis inhibitors, and this was quite hot probably 5 or so years ago. Judah Volkmann, who is sort of one of the heroes in cancer and a very interesting person, and there's been some interesting scientific biographies written about him, had this idea, and it was backed up by data, that I'm trying also to advance the slide, but I'll talk as we go, that cancer cells need a blood supply to actually form and develop, and that actually cancer cells send out special signals to induce new blood growth, he discovered, and that those new blood vessels both provide nourishment for the cancer cell and also provide the vehicle by which they can spread. So he said, if we could just get rid of this signal that's inducing new blood supply, we can kill the cancer. And we might even have almost a magic bullet, because this is a property of virtually all solid tumors. And it was very exciting, and some people thought we were really onto it. And then, as most things happen, here's sort of a picture of some of it. But as more and more trials were done, it seemed as though, although there was some advantage to it, usually it needed to be done in combination, and it often was not effective. So, for example, pertuzumab, which is bevacizumab, which I'm not pronouncing too well, which is also better known and easier pronounced to call it Avastin, a Genentech drug, was approved in some cancers and actually had some efficacy. It got accelerated approval in breast cancer, in fact. And then when the studies were completed, it turned out that it actually was not adding any benefit, and it was actually fairly toxic, because it hit blood vessels not only that were sent out by cancer cells, but others. And this is a place where rash was a really very difficult toxicity, and we all have some rashes from mosquito bites and dry skin, but some of the rashes caused by cancer drugs are really very, very fast. So I'm not talking really in synchrony with the slides, because they're not keeping up with my clicking. But I guess here we just see this bevacizumab and Avastin example, and it binds to these receptors and is meant to kill the blood vessels. There are some successful anti-angiogenesis treatments now that have been approved, and I don't think there are any blockbusters here, but they have been approved in a number of cancers, and there's still research going on. But I think the hope that this was going to be a magic bullet is definitely not been realized. So we're going to now turn to immune therapy, and this is a topic that has been, sort of on. Well, first of all, it was a hope. Many, many years ago, before even chemotherapy became really hot, that somehow or another we could use our own immune system to kill cancer. And it just seemed like, it was such a natural, easy way. We wouldn't have the toxicities, slam dunk, but it really took many, many years for anything to happen. There were a lot of disappointments. A few times when some cancers definitely have a viral component, and there was maybe some hope, but lots of work that didn't go anywhere. But I'd say 5 or so years ago, when you started going to AACR, you would hear some promising stories, both in terms of the science and in terms of the clinical work. And in the last 2 years, it's phenomenal, and there are now treatments that are really almost, some people almost use the C word for that is cure, not cancer. So I'm going to tell you a little bit about some of them if I can get the slides to move. But, I guess we didn't do the bottom line, I'm sorry. So I think I said all these things. If I didn't, and the expense, this is another one of those Genentech blockbusters that cost over $10,000 a month, I believe. That's a personal comment, sorry. Immune therapy, the idea is that we can somehow strengthen the immune system to produce anti-cancer activity. And there are a number of different approaches to it. Actually, the one that is most exciting isn't even listed here, but I will talk about it some. The immune system, which we probably all learned a little bit about in high school biology, is extremely complex. I've been trying to learn a little bit about it, but it's more complex, and it has more different kinds of things than when you learn about genomics, at least I can assimilate some of that information. The immune system is just so complex, and it's going to get more complex before we understand it. But what it's doing is it's protecting us from things that invade our bodies. And it's amazingly developed. There are multiple systems, an innate system and an adaptive system. It has a lot of specificity. It can have some memory, and it's really amazing. But what is really important, and it has all kinds of feedback loops and checks and balances, what we need to do is have a fine line between going after things that are foreign to the body and killing them, like bacteria or virus or whatever, versus killing ourselves, or our own cells, which is what autoimmune diseases do, where it starts, trying to kill things that really are us, even if it's a little different. Now, cancer is right in the middle of that. Cancer is us, but it's abnormal us, and the immune system probably kills some cancers, and it's one reason why we don't all have lots of cancers, because we all have lots of mistakes in our cell development. But it misses some, and the theory here is, what can we do to, get it back fighting the cancer, but not causing autoimmune diseases. So this is a picture that shows the things that we probably learned about in high school, about all the different organs involved, and the white blood cells, which are sort of the key actors in the immune system, the workhorses, as it says here. And this is showing a picture of a white blood cell, probably in the innate immune system, going and sucking up something that doesn't belong there. So we talked a little bit before about antigens and antibodies, and antigens are the things that the immune system recognizes, and antibodies are things that the immune system produces, and it hooks up. It's sort of a lock and key system, so that it inactivates the antigens, the innate immune system has these things all around, looking for things that don't belong and sort of eating them up. But in addition, there's an adaptive system that can learn to recognize things that don't belong and start creating antibodies to kill them. And there's a lot of checks and balances, so it doesn't get over control. But that's how many vaccines work. So when we get a smallpox or a chickenpox vaccine, they put in something that's very similar to the smallpox or chickenpox, and that's not active. And then our immune system sees that and starts producing things that would hook into that and recognize this foreign thing. And if a real chickenpox or smallpox cell comes, it would go after it. And when I said before that the adaptive immune system has memory, that's what I mean. Once you have these antibodies, they usually stay in your system. Although sometimes if they're never used, they don't, and in some cases we need to get boosters. Before we go to cancer vaccines, I want to talk about what are probably the most exciting new types of immune therapy, and they're what's called checkpoint inhibitors. And we don't have pictures for this, but there's some really nice stuff on the web. If you go to, I can give you some sites that have some little, video stuff and training stuff that you can learn more about. But what happened is that cancer cells actually send out signals. They're very sneaky cells. They send out signals to recruit blood cells. They also send out signals to tell the immune system not to kill them. These are called checkpoints. So there are a whole number of them, and there are 2 of them that now have approved drugs, and others that are in research. So the cancer cell sends out these signals that say, oh, immune system, don't get me, and then the immune system doesn't get it. And we now have some inhibitors for a checkpoint called CTLA-4. The drug is called ipilimumab, and in 2011 it was approved by the FDA on a rapid approval. It's a drug produced by Bristol Myers Squibb, and it has amazing effects in a significant proportion of melanoma, which up to that point had really a very poor prognosis. And the reason that I'm spending time talking about these checkpoint inhibitors is because although they don't work in all patients, whom they work in often have very long-term responses, what's called durable responses. And another thing that's really interesting about them is sometimes, even if you don't see a progression-free survival advantage, you often do see an overall survival advantage, which is sort of the opposite of many chemotherapies or other targeted therapies, and overall survival is what we need. And the theory is that even if the immune system isn't managing to totally fight back the tumor growth, it's slowing it down. So there's continuous growth, but perhaps at a slower rate. Some of these have side effects, but many of them don't have too bad side effects. In addition to the CTLA-4 inhibitor, in 2014, both Merck and Bristol Myers Squibb came out with PD-1 inhibitors, which actually are related to the T-cell receptors. And I'm getting beyond anything I know, so I won't say much more. but the drugs are called pembrolizumab and nivolumab, or something like that. And they were both approved just last year for melanoma, and just this year, the BMS drug was approved for non-small cell lung cancer. And there are many, many, many trials now going on in virtually every solid tumor. There are some indications that hypermutated tumors, which typically melanoma are more susceptible to this, because there are these antigens on cancer cells called neoantigens that perhaps the immune system is recognizing, and more of them on mutated cells. There's also, with respect to PD-1 inhibitors, there's a marker that they can find in the tumor, PD-1 receptors, and people with very high PD-1 receptors are more likely to respond to these PD-1 inhibitors. But interestingly, it's good to have those kind of biomarkers, but in a number of studies, they find that even people who have very low PD-1 do respond, but at a lower rate than people that have high response. So, some of the trials are only going after people that have that marker, but it seems that for people that have no other option, there's a chance that this therapy may actually be helpful. Now, these therapies all are, these $10,000 a month things, and that's a whole other issue that I think we all need to think about. But it's amazing, and there are people that have been living for 5-10 years on some of these therapies that were expected not to live more than 6 months. Not everyone responds, as I said. In fact, probably on average, a larger proportion of people respond to targeted therapy, but those people that do respond have a much longer response. And if their tumor progresses and they go on something else, they can then come back and have this and have a second response to it. So, in addition to BMS and Merck, there's Genentech, AstraZeneca, MedImmune, Lilly. They're all experimenting with these, and several other types of checkpoint inhibitors, at least a half a dozen that I saw when I was starting to look at this. So I think by far that's the most exciting advance in cancer therapy and certainly in immune therapy. But I'm going to briefly go over a couple of other types of immune therapy. One other that there is some significant progress in, and others that I think less so. That is, if I can get the slide moving. So cancer vaccine, yes.
Dorothy asked if you could send the URLs for those sites that you mentioned.
Yeah, what I think I will do, if it's okay with you, Peggy, I will send them to you, and then you could forward them on.
I will.
Okay.
And Jim asked, he said our bodies produce cancer cells every single day, and essentially all of them are immediately eliminated by the natural killer cells. So Jane is right, the immune system is unbelievably complex.
Right. So I think, I don't know if everyone would agree that we're producing cancer cells every day, but maybe that we are. I think most people would agree that we are sometime doing that, and almost everybody is. We certainly are producing abnormal cells, cells that have mutations, and we have a lot of cell repair mechanisms and other things that get rid of those bad cells, not always the immune system, but many of, I think it's quite likely that many of them are taken care of by the immune system. So tumor cell vaccines can be made in a number of different ways. One is actually what's called autologous, from the patient's own tumor. And in this case, the patient's tumor is, some tumor is taken out, and their white blood cells are taken, some T cells are taken out, and they grow in a medium more and more of these killer T cells that are targeted to this specific tumor, patient's tumor, and then not implant them, but they reinfuse them. And there's probably about 1,000 patients who have now been treated with this type of therapy, to sort of jumpstart the immune system. And there's definitely been some success, but it's a very, very expensive type of therapy, even to do trials, because each individual patient has to have its own vaccine. On the other hand, allogenic vaccines people are experimenting with, where they take tumor tissue, but of the same type, but they take tumor tissue from a variety of different patients or tumor or cell lines that express a variety of different features, hoping that that will stimulate the immune system, and some of them will match the patients. And there's a lot of work in a lot of different types of cancer to try to develop those. There have been a couple of vaccines that have been approved, but only a couple. And then there are some that try to get at the dendritic cells, which is another important part of the immune system that I can't say I really understand, but I think the dendritic cells are believed to sort of show the antigen to the immune system, and the killer T cells are the ones that go and actually kill it. So I think I better stop before I get myself in trouble. We talked about monoclonal antibodies already. As I said, I think these the monoclonal antibodies that go after receptors that inhibit the immune system, I would call immune therapy. The ones that go after other kinds of receptors, like the HER2 receptor, I don't actually, although they're monoclonal antibodies, I don't consider them an immune therapy, but I know that there's differences of opinion on that. So let's see if we can move along. This slide actually shows you a number of the drugs that have been approved that are monoclonal antibodies, and we've already talked about a number of them. So the bottom line is that potential advantages: many new monoclonal antibodies have recently been approved, seem to extend survival. Herceptin, which is, I don't consider an immune therapy, but as a monoclonal antibody, has had dramatic effects for, about 25 or 30% of breast cancer patients who overexpress HER2. The checkpoint inhibitors have already made a huge difference in melanoma and are starting to make a significant difference in other solid tumors. The disadvantages, at least with respect to vaccines, is they've had limited effects and they're extremely high cost. Some have significant severe side effects. As I said, many of the immune therapies are better at slowing the growth than actually eliminating the tumor. But I think as a patient, that might be something I would be more than happy to trade off, although if you have a very bulky tumor, that can really end up being very symptomatic. So I think now we're going to get to slides on gene therapy, which is another area that has been of potential interest and excitement to actually manipulate the genes. But in truth, there have been very, very few, if any, real successes. I don't think there's any gene therapy that has been approved for cancer, although there are some in other diseases. If the slides move on, we can talk about it. There is like a little Genomics 101 that's in the slides, and you'll get the slide deck. but I suspect this audience kind of knows a little bit, not as much as is on this slide, about genomics. We already talked about cancer being abnormalities in the gene, but there can be a whole bunch of different types. The problem with gene repair is something that I was mentioning in response to Jim's question, where when we make mistakes, we have built-in mechanisms that are not part of the immune system, but also the immune system. And epigenetics are a sort of a system on top of the genetics that are things that are not inherited, but actually turn genes on and off. And there are now some new therapies that I think at least one has been approved, but there's quite a bit of research going on, and it looks like you can actually turn some of these signals on and off and make them more, or then make the cancer cell more or less sensitive to some of the other therapies. So they're usually used in combination, but not given at the exact same time. Like immune therapy, there are a lot of different approaches that people have thought about and tried in respect to gene therapy, inserting genes to replace a non-functioning gene, swapping a gene, repairing a malfunctioning gene, altering the regulation. And there are technologies that can be used to do this. So the viruses that actually invade the genome can be manipulated to get rid of the gene, the virus-causing part of it, and use it as like a vehicle in which you can insert the gene that you want to deliver to the patient. There's some risk with this. There have been some errors or some tragedies associated with it, but it's something that's still being tried, but hasn't gotten very far. Liposomes are nanoparticles that have sort of round, fatty things that can carry genetic information to the patient, and I don't know that there's been much on gene manipulation using liposomes, but they actually are used to deliver some chemotherapies in potentially less toxic ways. In particularly, Abraxane is a form of Taxol that's in this kind of delivery mechanism. And what it allows you to do is either have less toxicity or keep perhaps the same toxicity, but increase the dose and hopefully have better efficacy. And that trade-off is not totally understood. The drug itself is approved for, I believe, pancreatic cancer and is being used off-label and in trials in other cancers. Actually, there's a belief that we shouldn't be using steroids with the new immune therapies. This is an aside, by the way, because steroids really impact inflammation, which is related to the immune system. So many of the trials that are doing in combination Taxol with a immune therapy are being done with Abraxane, which is much more expensive than Taxol, but has this property of the liposome means that you don't have as much of the gut types of nausea and things, and less likely to need a steroid. Although that in theory is the case, I've been hearing lately that in practice steroids don't really interfere with the therapy, and sometimes you need to give them anyway. So that's an open question, and if you ever are reviewing a concept that is talking about the checkpoint inhibitors, that might be a question that you would want to ask. So let's see. These are the ways that the gene could be manipulated, and I think what we have now are just some pictures of it. Stem cells, there's lots of stuff going on with stem cells, both general stem cells that could be programmed, and also cancer stem cells, which is a whole other topic that I personally think is quite interesting, probably because one of the researchers at Michigan, who I am very fond of, is doing it, is working on cancer stem cells and believing that a lot of traditional therapies don't work because we kill the cells that are not stem cells, and the stem cells are resistant to many of these therapies, and then go on to, you can have a reduced tumor, but the stem cells go on and make it more. Let's see where we are now. So here we have the bottom line for gene therapy is that the idea is to make cancer cells more sensitive to other treatments, or to make healthy cells more resistant to side effects. They can cause the suicide of cells, and that's a real problem. Very high cost, many unanticipated side effects, including some very well publicized deaths. May have impact on descendants if we're actually manipulating the genes, and any hint of a positive impact might only be short-term, and to my knowledge, this is an area that has not been moving very far, very fast, and I suspect that in a number of years it may, but at this point it's not. So I think we're sort of getting to the end, and I'll do some summary, and then maybe we'll get to some discussion and questions. New treatments come from research, as we know. We're all about facilitating research, and in particular the clinical trials, which our responsibility is to think about them in terms of the patient. These trials that patients will want to get into, these trials that are treating the patient as well as possible, both as an individual by respecting their time and taking care of it being convenient for them, but are also, really trying to have an impact on the patient who's volunteering as well as the future. Is the science that people are going after really going to make a difference? We, as an advocate, I think the important thing is to keep asking questions. Why did you do it this way? Why did you use that drug? Why did you use this dose? Have you thought about this other thing? I think you'll have trouble recruiting because patients will not want to be randomized, or they won't want to. You should consider having a 2-to-1 randomization instead of a regular randomization. You can also talk to Jim O'Malley about some other designs that could be more attractive to patients. Perhaps we should make sure there's crossover, so patients who don't get the experimental therapy have the option of getting it if they need it. So there are a lot of, as you know, patient-focused questions that we need to ask, and the more we know about the science, the more confident and competent we are at asking questions. But it's always we only need to know enough to ask questions rather than tell the researchers, and we only need to know enough to really think about this from the patient's perspective. So some points about targeted therapy here, and the bottom line, we maybe have made progress on the war on cancer, but it's not going away so fast. So with that, I think I will encourage more questions, more